All right, Krebs cycle more in depth. Now I'm gonna discuss more of the major players involved in just the Krebs cycle that I did not go over in the previous video. Go back and watch those videos in order to understand the yields that the Krebs cycle produce. I'm just gonna go and explain more terminology that's involved and what is produced in the Krebs cycle. So we're gonna get into the lettering convention. And the lettering convention goes as follows. C, C, D, alpha, S, S, F, M, O. Now, I'm going to discuss what each of these mean or what each of these stand for. So, I've drawn out every letter in this circle because this is the Krebs cycle. So, get straight into it. Citrate. Now, citrate is the weak acid that is in your citric or in your citrus fruits. And it is in just about every aerobic organism. And it serves as an intermediate for the citric acid cycle or the Krebs cycle, if you will. Those terms, again, are interchangeable. Citric acid cycle, Krebs cycle, same thing. All right, after citrate is formed, water is yielded. Then we come on over to our second C, cis aconitate. Now, cis aconitate basically takes citrate and rearranges it and turns it into an isomer. And after cis aconitate does this thing, more water is yielded. And once cis aconitate finishes citrate, it turns citrate into deisocitrate. And this term basically means that citrate is now an isomer of its former self, which means that it's made of the same stuff, but chemically it's been rearranged in order to accommodate the Krebs cycle, or in this case, to accommodate the Krebs cycle. So after deisocitrate is formed, we're going to yield off our first NADH or NADHH positive. And we're also going to break off our first carbon dioxide. Or I'm sorry, we're just going to break off our first carbon to form carbon dioxide. And then from there, a dehydrogenase is going to act on this isocitrate called deisocitrate dehydrogenase. And that's going to break it down, and then it's going to go off into our first alpha. I'm just going to write, because I don't have very much room, deisocitrate dehydrogenase. So I'm just going to call it dehydro for short, because I'm running out of room. Now, the first alpha stands for alpha ketoglumerate. Now, this chemical is one of the few keto agents that are involved in the Krebs cycle. I'm going to talk more about this in another video, so for the sake of simplicity, just know that it is a keto agent, or at least one of the few keto agents involved. So, after this is made, we're going to get our second NADH, or NADH plus, to be more specific. We're going to break off another carbon to make CO2. And just to make sure, collect my thoughts, make sure I'm not missing anything, another dehydrogenase is going to act upon it, called alpha ketoglumerate dehydrogenase. So we're just going to write it shorthand. Alpha keto dehydro. And again, that's shorthand for alpha ketoglutarate dehydrogenase. Ooh, I butchered that, but that's basically what it is. Alpha ketoglutarate dehydrogenase. That is a tongue twister. Please forgive me for that. Now we're gonna come to the first S. Succinyl. Make sure I spell that properly. S-U-C-C-I-N-Y-L, CoA. Now, succinyl-CoA is responsible for producing our first and only ATP of the Krebs cycle. You have ADP, then it's gonna add a phosphate group, making it ATP, and then another enzyme is going to come in and catabolize that, called succinyl. I'm going to write it shorthand. Succinyl-CoA synthetase. So a little bit of a curveball there. Now, why it's called that, I don't know. But it's succinyl-CoA synthetase, which means that this is a coenzyme. And that coenzyme helps produces ADP and ATP. Now, we go to the second S, succinate. Now, succinate is responsible for brain fart. Succinate is responsible for the production, or it leads into the production of our first and only FAD. To FADH plus. Now we're going to come on over to our first F. Fumarate. Now, fumarate is a salt and ester, and then another enzyme is going to come in. You see the pattern here? Whenever there's a major substance, an enzyme comes in and acts upon it. Fumarase. It's going to catabolize that, and that's going to lead into malate. Oh, just a quick side note. Anything ending in ASE is likely an enzyme. It's going to be catabolizing something. And when something is catabolized or being catabolized, that means that something is being broken down chemically. Then we have malate. Now, malate is another salt ester that leads into our next and final NAD to NADH, or NAD to NADH plus. Now, we come full circle to oxaloacetate. Now, if you remember, we lost 2 carbons here. So we come back full circle to a 4-carbon oxaloacetate. And if you remember, this is the substance that combines with acetyl-CoA to make citric acid. And it just brings full circle just like that. And then after oxaloacetate is formed, the final product here is water. All right, CC-FOMO, cis-aconitate, deisocitrate, alpha ketoglumerate, succinic-CoA, succinate, fumarate, malate, and oxaloacetate. This is the Krebs cycle more in depth. Now, go back and watch my previous videos and you'll have a better understanding of all the definitions of these terms and what NAD to FAD to ADP and ATP, all of that is discussed in previous videos. I hope you learned something. Keep hanging 10 and I'll have more content for you.